Welcome to this episode of PharmTech's Pharma Fundamentals. In this episode, Robert Dream, Managing Director of HDR Company and a member of PharmTech's Editorial Advisory Board, provides an introduction to an explanation of hold times for biopharmaceuticals.
I am Robert Dream. I've been in the industry for close to 40 years, working basically in different sectors of the industry. I am a consultant, HDR Company LLC. Today, we will basically cover the hold time in biomanufacturing and do some kind of comparison to small molecule as well. So hold time in biomanufacturing is the maximum allowable period during which a material can remain at a defined stage of the manufacturing process before moving into the next processing steps without adversely affecting its quality, safety, purity, potency, and stability. So hold times are established through scientific studies known as hold time validations or hold time studies. These studies demonstrate that the material remains within a predefined quality specification throughout the approved holding period. Let's say, what is a hold time? A hold time is a controlled waiting period between two manufacturing operations. Rather than immediately proceeding from one processing step to the next, manufacturing may intentionally or sometime unavoidably pause production because of equipment availability, quality control testing, scheduling constraint, cleaning and sterilization, batch release decisions, transportations between manufacturing studies, suite, facility maintenance. And during this pause, the intermediate material must continue to meet all quality requirements. Why are hold times necessary? Biopharmaceutical manufacturing rarely occurs as a one continuous process. Defined operations may occur hours apart, overnight, several days apart, at different manufacturing facilities. So therefore, intermediate material often requires temporary storage. Without scientifically established hold times, manufacturing control demonstrate that the product remains suitable for further processing. During a hold time, the material is not actively processed, but remains under tightly controlled conditions. These conditions may include refrigeration, for example, 2 to 8 degrees C, frozen storage, -20 or -80, depending on what you're storing, room temperature storage, if basically not important to keep it cool or frozen, controlled mixing, nitrogen blanket to avoid oxidation, sterile closed containers, light protection, some products are light sensitive, controlled humidity, The exact storage conditions depend on the product and manufacturing processes. It's not uniform across the board. A drug substance is the active pharmaceutical ingredient produced before formulation into the final medicine. Examples include monoclonal antibodies, recombinant proteins, vaccines, cell therapy products, gene therapy vectors, enzymes. During manufacturing of the drug substance, hold time commonly occurs between purification steps. Each hold must be validated independently. Also, need to be said that if you basically have a hold time and the protein that you are dealing with is not stable for a long time, you really have to add those hold times. It's not just the hold times between the step, but you have to be careful of the total hold time from the beginning of the process to the end of the process until it's in a unit dose for the patient. Hold times for drug product. A drug product is the finished medicinal product that will ultimately be administered to the patient. Examples: injectable solutions, vaccine vials, infusion bags. Hold times may occur after formulation, but before sterile filling. What is monitored during a hold time? Control the temperature, prevent degradation or microbial growth, control time, ensure validated hold period is not exceeded, sterility, bioburden, prevents microbial contamination, pH, detects chemical instability, protein concentration, confirms product consistency, appearance, detect precipitation, dissolution, discoloration, or particles basically appearing in the product. Potency, ensure biological activity is maintained, because over time you can degrade proteins that mean your product is being degraded. Purity, defects, degradation products, or micro impurities. How are hold times established? Hold times are not chosen arbitrarily. They are established through control studies. For example, you do a study at T0 as starting time, 12 hours later, 24 hours later, 24 hours later, 72 hours later, 96 hours later, as an example. And if your testing basically of the product passes through 72 and fails at 96, that means your hold time maximum can be 72. For a safety factor, usually manufacturers take less than the maximum, because the maximum usually is not given 100%. It could be at a different time, different change in parameter might not be exact. Hold time in biomanufacturing differ significantly from those in small molecule pharmaceutical manufacturing, because the product themselves are fundamentally different in their composition complexity, stability, and sensitivity. Environmental conditions, while both industries establish validated hold time to ensure product quality, during manufacturing, the scientific basis, risk and control strategies are much more demanding for biological manufacturing or biomanufacturing. Nature of the product. one thing to be considered. The most important distinction lies in the type of material being manufactured. Biopharmaceutical are product using living system, such as mammalian cells, bacteria, yeast, or other biological organisms. The resulting product are typically large, complex molecules, including monoclonal antibodies, recombinant proteins, vaccines, enzymes, cell therapies, and gene therapies. Small molecule drugs are chemically synthesized compounds with relatively simple and well-defined molecular structure. They are generally much smaller, more stable, and less sensitive to changes in their environment. So you can see the difference between the two. There is a lot more difficult to establish a whole time for biologic than small molecules. Greater sensitivity to degradation. Biopharmaceutical products are highly susceptible to physical, chemical, and biological degradation. During storage between manufacturing steps, potential changes include protein unfolding or denaturation, aggregation of protein molecules, fragmentation, oxidation, deamidation, loss of biological activity, formation of visible or subvisible particles. Even relatively small changes in temperature, pH, agitation, or storage duration may alter the structure of biologic and reduce its effectiveness or increase its immunogenicity. In contrast, many small molecule drugs remain chemically stable for much longer periods and are less likely to undergo rapid degradation under normal manufacturing conditions. Higher risk of microbial contamination. Biopharmaceutical manufacturing frequently involves aqueous water-based solutions that are rich in protein, nutrient, and other biological materials. These environments can support microbial growth if contamination occurs. Consequently, hold times for biologic must carefully be controlled. That are sterility, bioburden, endotoxin levels, container integrity, environmental conditions. Temperature plays a much larger role. Temperature is on the most critical factors influencing hold times in biopharmaceutical manufacturing. Depending on the product, intermediate may require storage at controlled room temperature, 2 to 8 degrees refrigerated, minus 20 degrees C, minus 80 degrees C. Even a brief exposure to temperature outside validated ranges can accelerate degradation or reduce product potency. Many small molecule intermediates are considerably more temperature tolerant and may be stored safely at almost ambient conditions for extended period. Biological activity must be preserved. For biologics, maintaining the correct molecular structure is not sufficient. The product must also remain its biological function. During hold time studies, manufacturers often evaluate biological potency, binding activity, enzymatic activity, cell-based functional assays. These specialized tests demonstrate that the product continues to perform its intended therapeutic function after storage. So it's really important to keep these basically activities in control. More complex quality attributes, which is like when you have a biological drug product, if the quality attribute changes, really the drug is no longer good for the patient. Biopharmaceutical products are evaluated using numerous critical attributes, CQAs, many of which can change during a hold period. Typical CQA include protein concentration, purity, aggregate levels, glycosylation profile, charge variance, molecular integrity, potency, sterility, particulate matter. So those are some of the CQAs basically you keep an eye for. A change in any of these characteristics may affect product safety or efficacy. Hold times studies are often more extensive. Because biologics are more sensitive, hold time validation studies generally require more comprehensive analytical testing. Scientists might evaluate samples at multiple time points to determine whether storage affects product quality. Typical analysis may include purity testing, potency assays, protein aggregation analysis, size exclusion chromatography, electrophoresis, charge variant analysis, sterility testing, bioburden testing, pH measurement, appearance assessment. Those are some of the testing basically scientists do during the validation of the hold time. Small molecule hold time studies often emphasize chemical stability, impurity formation, assay, and moisture content rather than complex biological characteristics. So you see the difference. There's a lot of difference between the two. Manufacturing processes are more interconnected. Biopharmaceutical manufacturing consists of multiple highly integrated steps, including cell culture, harvest, clarification, chromatography, purification, viral inactivation, viral filtration, ultrafiltration, diafiltration, formulation, sterility filtration, and filling. Delays at the one stage can influence downstream processing because the biological material continues to change over time. As we said, is the hold time basically is not between the steps, as the total hold time is important. You have to basically, you could go between 4 or 5 steps and you are okay, but then your proteins start degrade or whatever you are processing. Small molecule manufacturing generally involves sequential chemical reactions, purification, crystallization, drying, milling, and formulation. Although delays may affect manufacturing efficiency, intermediate are often more robust during temperature storage. So it's like a lot easier to manufacture. Product type. usually large, complex biological molecules, and small molecule. The small molecule means what it means is small chemical compounds. Stability, generally less stable. The small molecule generally more stable. Temperature sensitivity is high for biologic, moderate or low for small molecule. Microbial risk, high due to aqueous biological material, usually lower for small molecule. Potency concern. Biological activity must preserved for biologic, primarily chemical potency for small molecule. Quality testing, extensive biological, biochemical and biological testing, primarily chemical analysis only for small molecule. Hold time limits, often shorter and tightly controlled for biologic, often longer and more flexible for small molecule. Storage conditions, strictly controlled, temperature, sterility, mixing, typically less restrictive for small molecule, depending on the process. Are there different types of hold times? So yes, in biopharmaceutical manufacturing, there are several different types of hold times, each corresponding to a particular stage of the manufacturing process or a specific material being temporarily stored before the next operation. Although all hold times serves the same overall purpose, ensuring that material remains safe, stable, and suitable for further processing, they differ in what being held. Where in the process the hold occurs, the risk involves the storage condition required and quality attributes that must be maintained. Understanding these different hold times is important because each stage of manufacturing present unique challenges. A hold that is acceptable for the intermediate may not be suitable for another due to different in product stability, sterility requirement, or a process sensitivity. So process intermediate hold times is one of the items that basically while you are processing, you have to hold the intermediate. A process intermediate hold time refers to the maximum period that an intermediate material may remain between two manufacturing operations before further processing. An intermediate is partially processed material that has not been yet become the final drug product or drug substance. Example include harvested cell culture fluids, clarified harvest, chromatography pools, viral inactivation pools, buffer exchange protein solutions. These materials are often held while waiting for equipment availability, process scheduling, quality control reviews, operator availability, batch coordination. Characteristic occurs during active manufacturing, usually involves liquid biological materials. Storage conditions are highly controlled. Quality must remain unchanged throughout the validated hold times. Drug substance hold time. Drug substance hold time refers to the product during which the active pharmaceutical ingredient can be stored after manufacture, but before formulation, into the finished drug product. Examples, purified monoclonal antibodies, recombinant protein, vaccine bulk, concentrate, and a drug substance are commonly stored at refrigerated, frozen, in sterile storage vessels, in frozen bags or bottles. The objective is to preserve purity, potency, structural integrity, stability. Drug substance hold times are particularly important because degradation and this stage may directly affect the quality of every finished batch manufactured from that material. Drug product hold times. Drug product hold times applies after the active ingredient has been formulated into its final pharmaceutical preparation, but before subsequent manufacturing steps, such as sterile filtration, filling, or packaging. Examples include formulated vaccine solution, injectable solution before filling. During this hold, manufacturers monitor sterility, appearance, pH, potency, protein stability, particulate formulation, because the formulation is closer to the final patient product. Maintaining product quality is especially critical. Bulk hold times, again, this is basically the product is in a bulk, not formulated. Examples are bulk drug substance, bulk formulated product, bulk vaccine subsequence, and basically you're dealing with the same parameters as far as bulk material may remain in different conditions, and the whole time often necessary while manufacturing awaits. Pre-filtration hold times, that's kind of well known. You want to basically kind of limit microbial growth, protein aggregation, chemical degradation, increased bioburden, etc. Post-filtration hold times. This is one of the most critical hold period because the product is considered sterile. Any contamination introduced after filtration cannot usually be removed, and extended storage may compromise the sterility assurance of the product. Buffer hold times. Manufacturing buffers are used throughout purification, filtration, and formulation processes. Examples are equilibrium buffer, wash buffers, elution buffers, formulation buffers, and hold time include pH stability, microbial contamination, conductivity, and chemical composition. And the last one is media hold times. Cell culture media are prepared before inoculation, product inoculating into bioreactors. Prepared media may require temporary storage before use. Scientists evaluate nutrient stability, sterility, pH, and osmolality. Cleaning hold times, that basically cleaning facility, cleaning equipment, cleaning utensil, all that kind of stuff. And you want to make sure basically after you clean them, you kind of create a hold time for them before they get contaminated. Dirty hold times, again, similar to the cleaning hold times. You want to basically create a hold time for dirty because stuff can bake on the equipment, can create a lot of difficulties to remove it. You can create enormous growth that is not easy to clean. So dirty equipment, dirty utensil, dirty facility, it has to have a hold time, how long you can hold it before you clean it. So the other thing is the sample hold times and has exactly the same thing as the product hold times because your samples has to remain basically as they taken.
Thank you for watching this episode of Pharma Fundamentals. Visit pharmtech.com for more video episodes and articles in this series.